My name is Donna Peer. I'm now at the Sloan Kettering Institute. And I'm about to give you a talk that I tried to balance a little bit of, I wouldn't call it technical details as much as technical insight with a little bit of material. So we'll see how this mishmash goes. You tell me. Before we actually dig in, I want to make a little plug for the Human Cell Atlas project. In October of this year, I'm supposed to stay against the wall, I forgot. Yeah, in October of this year, we launched the Human Cell Atlas project. This project is different than the genome project in that the genome really wanted to define the genes and the DNA units. Here, the cell becomes the central unit. We need to define our cells, what they do, where they are, how they're organized spatially, how they form through the process of development, how do they switch states. And we really want to understand all that. Towards this goal, we need a lot of computational people. I lead the computational analysis group along with John Marioni, so I invite anyone and everyone to help all the computational needs and infrastructure of this huge project, likely to be a 15-year project. And we need computer science for scale, scope, and computability of the scale of this project. Now, really, what made this project possible was the rise of droplet-based, high-throughput, microfluidic, single-cell RNA sequencing. For those of you, how many people here know a little bit about this technology? Okay, about half. So let's go through it. The idea here is that we take cells, those are the little red thing, and we encapsulate them into these little droplets. Now we have a cell with a bead inside a droplet with different enzymes. And inside the droplet, the cell bursts, lysis occurs, so its RNA is released. And on these beads, there are primers. These primers have a cell-specific barcode, so that any read that we sequence will be identified to that particular cell, so we know where that read came from, and a random molecular tag so that we can count molecules. So after amplification and everything else, we know that this came from the same unique RNA molecule. So in the process of RT in these droplets, each molecule, each transcript gets barcoded with a random molecular tag, so we know its identity after it's amplified and where it came from. Now, in this microfluidic technology, we can capture thousands and tens of thousands of cells at once in a very cheap way. I'm going to show you a little, this is a microscope. See that little tiny thing in there? That's a cell coming in. There are cells, these beads coming in on one at a time, and RT enzymes. And this thing is slowed down about 100-fold, just so you know how fast this runs. This goes through about, this is one of the technologies, and it goes through about 10,000 cells in 10 minutes. Other technologies go quicker, but they're all based on this idea of really encapsulating a single cell inside a droplet and barcoding its transcripts so that we can now associate these transcripts back to the single cell. So one of the things that I've been playing around with for a while is everything you can do with single cell. But in particular, this basically RNA-seq based technology has both the wonder of being the entire genome as well as many technical difficulties. I was at the NIH 2 weeks ago, and some people there think that it's not even ready because of some of the caveats I'll tell you here. And the point is that the coverage isn't deep. We don't have the full transcriptome for every individual cell. What we have, depending on the technology and the depth of sequencing and the success of the experiment, about 2 to 10% of the transcripts in each individual cell. So here's one of the early papers, and they tried to show that two cells are highly correlated. And this is what I call a perfect example of misleading visuals by the Broad. Let me change this scatter plot into a density map. Okay, this is the same data, but based on density. where most of the data is on the zero axes, either on the x-axis or on the y-axis. And the small percentage of data that's creating that thing that looks correlated is a very tiny percent of your data. But the visual looks great on the left. Here's another example. This is another technology, not the Broad, though trained at the Broad. They didn't make this visual. They didn't want to show this visual. This is a human bone marrow. And what we have here are clusters, and there's enough transcripts in each cell so that even if we don't have the canonical markers by which these immune subsets, T cells, B cells, are identified, we can figure out, because there's so much distinct expression for each cell subset, which cell type each cell is. But when you look at the really canonical markers, the markers that immunologists like to look at when they say that this cell is a monocyte, or this cell is a B cell, or this cell is an effector T cell, they're really gone. Like all monocytes should have CD14, and here less than 2% have it. All of them should have CD11B, and here less than 6% have it. It's not that they're not expressing these transcripts. This is the sparse capture that we have here. And this particularly hits our ability to get gene-gene relations. I started in the field from the very early days as a young graduate student, being enamored by regulatory networks and reconstructing gene-gene interactions and gene regulation. But when you have such a high degree of dropout, your chance of getting two genes in the same cell are very low. So let's take this beautiful relationship, drop it out even less than the typical dropout that occurs, and here's what you have. Your ability to see that relationship is completely gone, and people have been complaining about the fact that you can't get these gene-gene relations out of single-cell RNA-seq data. In fact, that was the big complaint of the recent big Keystone conference in single-cell omics. Half the talks were whining about this problem with the gene-gene relations. So we came up with a fix. Smita, who was a postdoc in my lab, and now she's faculty at Yale, and she continued to work on the project from Yale, and David, a postdoc in my lab, and we call it magic. That might be a little bit of bragging, but that was my response to when I saw it working. I mean, it was a nice idea, but I didn't think it would work so well. Here's that same immune data I showed you earlier with the dropped-out read. So as you can see, most of the data is on the axes. These are the canonical markers that are used to define immune cell types, and every dot is a cell colored by its cluster. Again, we have all the genes to try and figure out its cluster, and most of it's on the bottom, most of it's on 0, and we run our magic algorithm. It's an imputation algorithm to learn from similar cells, and it imputes. You can see the cells are now organized in order, so each cluster, from the more naive clusters all the way to the more mature clusters, you see the branching and the bifurcation in the hematopoiesis as cells go and become new cell types. And this with no prior knowledge, just by learning information and sharing information between similar cells, reconstructed the entire hematopoiesis. Looking at the same data as before, after our imputation, it all gets filled up. And again, the structure is correct. It doesn't randomly fill up the wrong surface markers in the wrong type. It really preserves the known structures, the known gene-gene relations, and everything else. Now, in my field, particularly biologists are very, very queasy about the very idea of imputation. They're not queasy about all the mistakes they have in their reconstruction of a count matrix from cells. They aren't queasy about errors and noise, but they are queasy when you say the word imputation. How can you say that you think something's there if you didn't measure it? But fortunately, I'm talking to a very friendly audience where many of you are from the population and genetics community, and you guys have been using the imputation for a long time. And again, you're using the structure in your data for imputation. So in order to get imputation working properly, the only thing you need to do is figure out what is the structure of your data. What type of structure can you harness to get good imputation? And gene expression actually has a lot of structure from the very beginning, from the very first microarrays. The first thing anyone noticed, the first thing Mike Eisen noticed with the very first microarrays is, wow, these genes cluster. Gene expression works in module, lots of co-regulated genes. You have very few genes that are out there in the genome doing their own thing. And we could use this collinearity if we missed some of the genes in this collinear cluster, but not all of them, then again, this is a very strong structure in our data. Another more unique thing is the actual cell phenotypes, cell states, which cells are comfortable and happy and reside in, are actually reside on very structured, low-dimensional geometric shapes. What you see here is, again, hematopoiesis, but this is a different technology. This is CyTOF, so we have a good, accurate measurement for each of the 13 surface molecules measured. And using T-SNE dimensionality reduction to 3 dimensions, we can see that the data is structured. Each cell type, each cell here is colored by a different cell type, falls very nicely into very distinct shapes. Yeah?
Whenever I see this, I have a question. If you generated the same kind of plot just from the RNA-seq single cell data, will you get something similar?
Vanilla doing nothing to it?
No, I'm just saying, is that information in the RNA-seq?
If you work a little harder to overcome some of the noise and the sparsity of it, yes, we get the same thing in single-cell RNA-seq. But I wanted to sort of justify some of the methods which I extract this sparsity. And you could see that these things are anything. The other thing to notice is that they actually are pretty complex. They're not convex. They're anything. You have to see this sort of B cells twisting and turning in the green as they develop. They're not these nice, beautiful, well-behaving convex Gaussian balls as people like to model them before. So we have these complex, twisting and turning, low-dimensional shapes in which cell phenotypes reside. So these are the things that we're actually going to try and use. And I want to show one more thing again here. Notice that this is real data. You can see that the cells, as they develop, they twist and turn like this. And this is actually, we see this a lot. As markers rise and fall through the course of development, we have many of these sort of snaking turns in development, or this thing right here. And therefore, if we want to look at distance between cells, and we want that distance to be biologically meaningful, then we can't really take Euclidean distance. So if we take each cell as a point in high-dimensional space based on its measured phenotypes, for example, its measured gene expression, and if we just look at Euclidean distance in that space, we can get these two cells that seem close to each other because of this loop, but are actually pretty far away phenotypically, developmentally, and we really want to measure distances along the manifold of occupied dense cell types. And so that's exactly what we're going to do. And to do this, we actually embed single-cell data in a graph structure. So the idea here, and almost a lot of what I do is basically harnesses this graph structure. Each cell is a node in the graph. And now we connect cells to their most similar cells, to their K nearest neighbors. K could be adaptive, K could be changing. And this is the neighbor graph. Here we see a case again of the hematopoietic system. And you see that this neighbor graph is very modular. We see that each cell type creates its own little highly interconnected space of connected cells. Up on top, CD34 is a primitive sort of stem cell marker, and we see all the cells on top colored by that marker. And most of what matters is how do you build this matrix? How do you decide what the distance metric is, what K is? Building a good graph differs between the different applications, but the core idea of this graph, and now that I have a graph, while these big steps through space, these big Euclidean distances are bad because I don't know what unoccupied space I can go to and how I can jump really far from really interconnected things, if I actually take small steps within this manifold, if I take a small step between very similar cells and progress between one very similar cell to another, I remain within the manifold and actually can create a new distance metric. And here, a random walk through this graph between very similar cells, if I want to now get to two farther cells, is actually equivalent to diffusion. So what we do here is we take a graph based on distances, And here, actually, the initial distance process matters less. You could take your favorite Euclidean distance because you're only going to focus on a very nearby neighborhood. You're going to build a distance matrix and take a kernel in order to turn it into an affinity matrix. And choosing the right kernel, the right K, the right sigma really matters right here. But basically, now you have an affinity matrix. And I want to show you a little bit of why choosing a kernel, and particularly choosing the sigma or the distance matters. Here again is that twisty loop structure. And here's a kernel that you see. You have this off-diagonal bit right here, which is these sort of crossing these steps right here that we don't want. We want this on-diagonal structure right here. So as you choose kernels and you switch this to an affinity matrix, if you choose the kernel right, you really get that sort of one-dimensional line shape. But if your kernel's off, you actually sort of really now connect faraway cells. Here we have real data. This is actually the process of EMT. This is a different system. Every dot here is an edge between two similar cells. And as we power the matrix, as we raise the matrix more powers, that's like taking more steps in the graph. So this is all the steps in the graph of one edge away. By powering this matrix, I now get all cells that are connected by up to two edges. So now I can take a walk of length 2, and these are all the steps that I can get in a walk of length 2. And here is a walk of length 3. Now the point is that this is a weighted walk. This is the probability of reaching from cell 1 to cell 2. So I expand my neighborhood. In one hand, yes, I can get to more distant cells, but paths that are very likely, two cells that are connected through many paths in a very dense region, two cells that are very connected through here and here and here and here get a bigger weight, and cells that only have one path that are, might be outliers, they actually lower their weight. So while I can actually reach more cells by taking a longer walk, this graph is weighted such that I really pick up on the most likely paths and the dominant structures in the data. And actually, if I have an outlier step, it sort of lowers its weight with time. And so I just wanted to introduce you to the concept of diffusion maps. This was new to me a few years ago. We started playing this before we learned that these things existed, and then I read up on it and discovered, wow, there's an entire field of mathematicians that are studying these things, that are figuring out how to use these kernels, how to actually use these graphs, and how to use lots of mathematics to actually derive a lot of structures of these graphs. So one way to think of diffusion maps, and I did a lot of PCA in my life, is diffusion map is just like the eigenvectors, but on this agencity matrix. So rather than doing your PCA directly on the raw data, you are looking for the eigenvectors and eigenvalues on the adjacency matrix in itself. And this, rather than finding the strongest linear trends through the data, actually finds the strongest nonlinear trends of sort of densities. So it actually finds these, the second eigencomponent would be this long twisting line right here. So it really finds these sort of twisting and turning trends in your data. The eigenvalues explain the extent that each component is being used, or that each cell is relevant. So it's very similar to what you're really used to from PCA, anyone that's done a lot of PCA analysis. But it talks about the really core nonlinear structure of the data. The first diffusion component is the cell density of the data. So once we have this neighborhood metric, once we have, we've sort of learned the manifold, we've powered the matrix as many times as we have, and each cell has a weighted neighborhood of its most similar cells, it now basically uses these similar cells to learn what it might have missed out through simple matrix multiplication. So we take our noisy, sparse data matrix, we take our powered matrix of neighbors, and while we average cells from different neighborhoods, one of the really nice things is that each cell still remains unique here. Each cell still retains its own unique neighborhood with its own unique weights as we impute and get the imputed data right here, filling in a lot of the missing values by learning from nearby cells. So does it work? Problem is, all single-cell RNA-seq data has dropouts. So we sort of need to create synthetic data. We've created a few synthetic data sets. In this case, we're actually going to mimic single-cell RNA-seq using bulk worm data from the microarray. Who's heard of microarrays here? Oh, people still know what that is. I thought it was like those old CDs that people don't use it anymore. Okay, who's seen microarray data in their life? Okay, yeah. These are dying out things, but some old data sets have them. They're actually noisier than anything else. But the point is that we have a developmental trajectory here. And in bulk, we have sort of these filled out expression profiles. So we can take, this is like a gene along its developmental trajectory. These time points were taken during the course of development of this worm. And we can drop out the data, destroy the signal. This is actually only 80% dropout. And after magic, the signal is restored. I can bring you lots of these things, or I can get some basic statistics. It's particularly good at picking up temporal trends. It's also particularly good at picking up these gene-gene relations. So even if I drop out the gene-gene relations so that most of the signal is lost after I drop it out, I can still recover the gene-gene relations quite well. And this is when I only have 206 cells. It works much better when you have 3,000 cells, but then you have to have more weird synthetic data sets. Basically, you learn a structure and then drop it out. Okay, again, to show you how this process of diffusion is sort of filling out the data and learning structure. This is, we saw it earlier in 3D. Now it's 2D slices of that 3D monster I showed you earlier. So this is the raw data. As you can see, almost everything is on the XY axis. It's very rare that you get two genes that you care about measured in the same cell, even though they should be often in the same cell. If you sort of do, if you use only the first neighborhood, the distance 1, you begin to get things filled in, but it's still messy. T equals 3, you begin to get something that resembles structures, and these are the structures that match known biology. T equals 7, which is our optimum based on our criteria. You can see that you really get these beautiful nonlinear shapes that match the behavior of these pairs during hematopoiesis. Here's stem cell, this is EMT data. So again, a big, noisy, unstructured blob at the 0 axis. And after magic, structure forms in this data. Interesting here is we get a trajectory. In fact, what we've been doing time and time again is using the asynchronous nature of our data, either the asynchronous nature of development. And in our bone marrow right now, we have cells that are developing the entire time. So if I take a bit of my bone marrow, beyond being very painful, I'll have all cell types in all stages. Here, even though I take a cell line and hit it with TGF-beta to induce the epithelial to mesenchymal transition, this happens at different times and it's asynchronous, so I get, even at day 8, the epithelial state, the mesenchymal state, and everything in between. Here I wanted to talk a little bit about the importance of the kernel. And there's a lot of details I'm not going to go in about the kernel, but one of the most important detail I do want to stress is the fact that it needs to be an adaptive kernel. So you want to build a k-nearest neighbor graph. If we have the same sigma for all cells in our data, we have very dense regions where we have mature cell types. If we use sigma that works for there, well, then we'll get a disconnected graph. Our transitional cell populations are very sparse. Our primitive cell populations are very sparse. On the other hand, if we have a tiny sigma that worked well for our very rare cell populations, it will become very noisy in the main data. And so what we do is we do an adaptive kernel. And we actually try and and we also want all cells to have the same number of neighbors. Otherwise, these cells with lots of neighbors provide more information. If I have a cell and it has very, very, very many neighbors, its values are going to be transferred very often, more often than some sparse cell types, and they're going to dominate all my data once I begin powering the matrix. So when I do, basically the data collapses to sort of these very popular, frequent phenotypes. But if you do an adaptive kernel, where every single cell has the same number of cells, so we basically set sigma to be the distance of the kth neighbor, then you now have an adaptive kernel with a different sigma for each cell based on its local density. and then you can pick up these fine structures. Comparing to other methods for imputation, just to show you why magic worked better and what you need in order to get it to work. At the time we started working on this, I know there's quite a few imputation methods recently. There were two possibilities. Neither of them were originally developed for single-cell data. Everyone, when I started showing this, started screaming at me, but matrix completion is a solved thing. What are you reinventing the wheel? There's so many matrix completion methods in machine learning. For example, here's one of these methods, and it again completely messes up. It doesn't get the data well. And why do matrix completion methods for machine learning choose your pick fail? Because they trust the non-zero values. In machine learning, most of the matrix completion matrix basically say, we have a lot of missing values, we have a lot of zero entries, we want to impute them, but when we have a positive value, we actually trust that value. Dropout hits everything. Even if we see a value, even if we see a gene expressed, it probably is expressing more transcripts. Again, we're randomly sampling 5% of the transcriptome. We can pick up a particular gene, but we probably didn't pick up all its transcripts. So every single value in our count matrix is messed up. Every single value is missing more counts, and we can't just try and fill in those missing zeros, and we can't take methods for machine learning that do that. LRA is actually very similar in many ways to what we do, except LRA assumes linearity, and these single cell phenotypes, the sort of the shapes that I showed you earlier, are anything but nonlinear. And again, the assumption of linearity completely breaks in this data. So before magic, after magic, again, one of our original goals was to learn gene relations towards the goal of learning sort of actual networks from single cell data. Here's the data before. This is and afterwards you can see clear shapes. This is a blob. This is a temporal trend during the course of the epithelial to mesenchymal transition. More of these ugly blobs, structured biology. That's sort of really, we can now get the shape of these gene-gene relations. Of course, we can stare at these, and I can do picture shows of more shapes, but we want to quantify these shapes. We want to vectorize them so that we can compare them, so that we can quantify the information. And for this, again, we're going to use the KNN graph. We're going to actually look locally at each grid and figure out what is the local density. Again, density estimation in high dimensions is a disaster. But when you go to lower dimensions, when you actually do density estimation using the KNN graph and ask what is the distance of the kth cell, you can actually have some really nice and robust density estimation methods. And we can estimate the density and actually vectorize these plots. One more little trick that we do, single-cell RNA-seq data, if you actually do sort of these density estimations, there is really a dominant phenotype that dominates your structure. So if we want to understand the relationship between two genes, between two proteins, between two molecular components, we really want to condition. We want to look at the conditional distribution rather than the joint distribution. We want to know how molecule X influences the values and the expression and the behavior of molecule Y. Here's the same data exactly. I'm just going to condition. I'm going to do a conditional distribution instead of a joint distribution. And we have this, again, beautiful shape. So we can take the data. This is the data before magic, block. This is data after magic. We do two steps of vectorization to get a reliable sort of shape density vector for how our pair behaves. We do the conditioning so we can get it for the entire dynamic range. And this gives us a vector of 20 by 20. Now we can compare gene pairs. We can cluster gene pairs. We can do mutual information on gene pairs and quantify the strength of the relationship. And now here we can actually figure out the behavior of all genes during the course of EMT, including these intermediate steps. So this is a full map of the different trends and waves of genes and regulators during the course of this very complex, very important transition as genes begin to fall, rise, peak at different points, and finally the mesenchymal state. We picked up many new regulators that weren't known to be related to this, and we have a lot of ongoing, much longer validations that show that they are indeed involved and critical in this process. Many of these sort of intermediate steps were highly uncharacterized. One thing that we do know is we can actually pick up regulatory networks from this. So again, we can look at the information between a pair. When does a gene, when do we have a regulator possibly regulate a target? Well, the regulator should have information on the target. The regulator should have a sort of strong structure, and we have this sort of information score on this conditional thing. So we want a high information score. But information, it's correlative. It doesn't suggest regulation. But here we have this sort of temporal pseudo-time trend. So if it has high information and peaks after our regulator, taking these two criteria together, we say this is a putative target. And using this, we actually did a validation of an important yet uncharacterized factor because its chipped antibody doesn't work well. We did an overexpression versus of that particular ZEB1 factor. If we just use things that appear after ZEB alone, that's not enough. There's a lot of other factors involved. Just appearing after ZEB gives you almost no significance. This is based on a rank order. Based on DREAMY, our mutual information score on the conditional probability, you get some significance. So things that have high DREAMY tend to be influenced by ZEB1. We have some strength here. But when we combine the two criteria together, you really get a nice set of targets that shoot up really, really, really high after we overexpress ZEB. This is random pairs. This is where we are quite a few standard deviations away with a nice p-value to boost. And here's a couple other ones. So how much time do I have? 7 minutes, okay. So I'm gonna whiz through this very quickly. I didn't know how much I would have, but I wanted to add this at the end. One of the other things that we like to do other than learn relations is to actually learn the structure. So I'm really enamored by how a cell develops, this whole process. And what creates these manifolds is as cells actually transfer and develop and go down the sort of Waddington-like landscape, shaped by these regulatory networks, that's exactly what is creating this process in high dimension. So what we want to do is we want to do it the other way around. We want to take the single cell data, and from this high dimensional single cell data where we don't know this sort of branching structure of development and bifurcations, we actually want to learn the structure in place these cells and understand the developmental structure and the relationships between these cells. So our first attempt, I'm going to go through this very fast, was Wunderlist using just a linear simple transition. And here, again, we have this neighborhood graph, and we do distances based on these walks. And basically, we take shortest path, and that's the distance from a starting point. Of course, distances get messed up because you can sort of zigzag across these things, so we throw in a lot of waypoints. These waypoints allow us to locally refine the distance. The shorter you are to a waypoint, the more reliable you are. So we sort of refine by averaging to all these waypoints. And just to give you a little visualization of how the waypoints work, if I have a waypoint from here and a waypoint from here, it gets sort of twisted. So you see it behaves nicely here, but then as you get farther away, the distances get skewed. Here, as you get far away, this distance gets skewed. When you average across the waypoints, you have a very smooth transition and very good way of getting sort of your distance from origin. The other thing these waypoints are very good at doing is actually figuring out the structure of the data, finding these bifurcations. So again, now we have one of these graphs with the bifurcation, and we can compare the distance directly or through the waypoint, the distance to the waypoint, and then the distance from the waypoint to where we need to go. And if the waypoint is along the same branch, they should be more or less up to some noise similar. But if the waypoint is on a distance branch, then actually there's going to be a big disagreement in the distance. And if we take lots of cells, we can see lots of disagreements, and these disagreements have structures. Things on the sort of stem will all agree with each other and everything. Things on one branch will agree among themselves, and disagree with things in these branches, and this actually gives you these very clear structures. So the actual disagreement matrix, the actual disagreement between waypoints actually finds the bifurcating structure of the graph. Here we have two, but this works and generalizes to higher dimension. And you can even see this is the end, the strongest disagreement, as well as where things branch off. So really this graph structure and these bifurcations are directly encoded in this disagreement matrix. Here we're going to use diffusion maps to clean up the data. Again, diffusion maps really figure out, okay, these 2 points, they might be far away in Euclidean space, but there's lots of paths from here to here. These 2 points might be near, but it's a stretch, this very narrow bridge that you have to go through. So using diffusion maps really cleans up the data. It's very similar to the way we used PCA once upon a time to clean up noise in our data and take the smallest PCA components as sort of our data components, our real biology components. Here we run the diffusion maps and re-project our data into the top diffusion components and really get a very clean distance metric. Again, diffusion maps alone aren't good enough. They sort of are a little bit messy. You don't get the bifurcation. But once you have the diffusion maps to sort of get you the general structure, you could use these waypoints and these disagreements to get a very, very fine structure. And finally, the most fun thing is now we can actually say what is the probability of an individual cell getting to any one of the endpoints. So we want to take all the cell profiles from the early points. We want to build a trajectory, figure out each cell, how mature it is, how far along down in the development path it is. What are the branch ends? What are the different terminal fates we have? And what is the entropy? And for each cell, what is its probability of reaching each of these terminal end fates? So again, this diffusion map construction, here's the raw data. There's some structure. You can see some ends to it, but it's pretty ugly. You remap it on these top diffusion components. It pops the structure out. You run this sort of distance, and you can really get, here's the early cells, here are the later cells. And now you want to direct this graph. And to direct this graph, to build directionality in it, again, we can use the fact that we ordered the cells from the initial point, and we can sort of trim edges that go back too far. We're allowing the cells to go back a little bit, but not too far. So we have a directed graph. From this directed graph, we could actually find the endpoints. These are endpoints that have many incoming cells, and many incoming edges, but very few outgoing edges that have a very strong weight in stationary distribution. These are our endpoints. We identify them as the ends. They're also the end of the diffusion components. They're also at the tips of the fusion components. Once we have our terminal states, we can figure out now for each cell, and there's nice thing is that these Markov theories really give us a closed form way to figure out from each and every cell what is its probability to reach each and every one of these terminal states. And once we have that, we could also give an entropy for each cell, and we now have for each cell along what is the probability of this cell to reach each one of these 3 terminal states. And you can see that actually, even after you're a little bit past the branch point, there's a possibility to cross over. So, and there's a lot of evidence for the fact that actually the process of development is very fluid. Cells are very pliable. And even when they seem to be going down one path, this sort of Waddington landscape is pretty flat. And only at the very, very, very end do they commit. And even a cell down here can still cross over to here. But we have a really good way of mapping the probability and the fate probability of each and every cell from these single cell data. So to summarize, this is a great challenge that we have to sort of really map all cell types in our body as well as their regulatory and spatial relations, and I invite you to do that as part of the Human Cell Atlas. Also understand the regulation that arise, and graph-based geometry, basically mapping the cells on a graph based on their neighborhoods, and then bringing in all the wonderful tools that the fusion maps and ergodic theory and stochastic matrices and Markov matrices have from all these mathematicians. Every time we find a new paper and think, oh, we could use that, is a really powerful way to approach this problem. So I told you, mostly work at the beginning, magic is David and Smita. Manu has been really pushing this whole idea of bifurcating trajectories, a couple other wonderful people in my lab. And finally, I moved to Sloan Kettering. In fact, some of my affiliations here were still at Columbia. So of course, I'm looking for postdocs as always, but I have a few other positions available. We have a single-cell research center where there's actually stable staff positions available for someone who doesn't want the complete academic track, as well as all rank faculty positions. Thank you.